recording. So, Brumfeld is not here. Champion is not here. Gross is not here. Gunther is here. You got WBSU stuff on? All right. Credit. That's hard to tell in the dark. Randall is here. He was the first one here. Gets extra credit for that. Zion is not here. Jackson? Not here yet. Keys is here. If you're wearing WBSU stuff, let me know. Bez is here, and I can see yours. WBSU sweatshirt. Sigmund is not here yet. Slaughter is not here yet. Smith is here. WBSU stuff. Just take that as a no. Stover is here in WBSU gear. Tompkins is not here yet. The Nest is not here yet. Watson is not here yet. Young is not here yet. Zweiback is not here yet. Lawrence is not here yet. Wilson is not here yet. Pretty bad display so far. Kind of works out given what I'm about to announce. But I'm going to wait till 8:01. And Wilson is here. It's 8:01, so I'll close the door and we'll get started. As always, attendance is very mandatory. Participation is graded. If you're here, that means nothing to you. You don't have to do anything. But as usual, you can send me in the words of the attendance words for extra credit. If you're online, you have to send in those extra credit words to get any credit. It's not extra credit for you, excuse me. But if you're watching the video, as always, you'll have to send in the letter, the list of words and screenshots. So, and then of course, if you are here or online, if you're going to send those words in, they need to be done by 9 a.m. So, any questions about attendance? Okay, I mentioned this or reminded you guys the last time we met, and it's a good day to remind you again. Make sure you check your emails or your course announcements before you come in. I have a sick daughter. I almost was not able to come in again today. So had I not been able to, I would have posted that to the course announcements. And then you could have known, oh man, I better not come in today. So I do that for a reason. You know, I don't want you to come in and waste your time, Miles. So yeah, make sure you check your course announcements before you come in. Also, you should be checking your course announcements regularly anyway. So always good advice. So any questions about that? All right, some good news. Wanted to change things up for you, and since I don't ever get to do anything nice for you guys. So today's lab, it's going to be as usual. It's past due on Friday. The only thing I'm changing up on it is today, since it's a virtual lab anyway, you don't have to show up today if you don't want to. Just do it on your own. So after we're done with lecture today, you can just go do whatever it is that you normally would do on a day that it wasn't Wednesday. So any questions about that? Okay. I will be here and online. So if you do need me, you know, for lab, you can meet me here or online if you need me for lab. But again, you are not required to be here for lab today. So any questions? Makes sense. All right. Next week is a little bit different. Speaking of which, next week is going to be a team effort. So we're gonna we're looking at genetics. So we're gonna be looking at different phenotypes that I haven't taught you yet. So basically, I'm like, all right, everybody has blue eyes, raise your hand. And we'll count the number of people who have blue eyes. And everybody who doesn't have blue eyes, raise your hand. And we'll count those numbers, right? We're going to compare all these different things about us as a group. And we're going to use that to learn about genetics. So you have to be here next week. You'll get 0 credit if you don't show up next week. Or, you know, half credit, maybe jump through 7 extras, things like that. But anyway, yeah, to remind you, the pre-lab is due on Friday, as usual. This one has 1 set of video questions, followed by the introduction. And then questions about the introduction to prove that you read the introduction. So again, those answers should come from the introduction. Don't look them up. The idea is not just for you to learn it, but to prove that you read the introduction. And then there'll be a set of hypotheses and tables. I got a question before from somebody, which I'm glad they asked, because I want to pass this along to you guys. They said sometimes they have problems knowing when the pre-lab questions are over. 2 things I would say. First of all, read carefully. Like, for example, this one. When you're done with question number whatever, it goes to the last number and it says, Now keep reading and do everything highlighted in green, right? There's always something that tells you, but that's part one. Part two, don't tell me and don't wait till afterwards to tell me that. Let me help you. Like, I'm here to help. These aren't quizzes, these aren't exams. When you're working on something, you're like, man, where do I even stop? Shoot me an email, maybe online. I'm here to help you. So any questions about the pre-lab that's due on Friday? All right, again, I know I've been saying this a lot, but someone's still gonna screw this up. Exam 1, you can still retake it up until this Sunday. You can take it as many times as you want. The retake is open book. The retake is not timed. The only catch is you have to tell me when you're done, so I know to go adjust your grade, because your grade will be the average of your first grade and the highest grade that you got on your retakes. And without you telling me, I don't know, and your grade won't change. Does anybody have any questions about the retake? Okay, also this Sunday, your second exam is due. And again, we're gonna miss this. It's gonna be sad. But yeah, your exam for exam 2 is due this Sunday. Like I've been saying previously, if you need more than 50 minutes, you need to let me know so we can find a time to meet online and I can proctor your exam, in which case I don't care how long it takes you to take. As long as I know you're taking a closed book exam, you take pretty much as long as you want. So any questions about the exams? Okay, and then finally, one other thing. Once exam 2 is closed, then you can no longer retake exam 1. At that point, you can meet me online to get feedback. So go through all the answers and look at all your wrong answers, and I can give you the correct answer and explain why your answer was wrong and why this one is the correct answer. You're going to need that because the final exam is about 100 questions. The first 50 questions comes directly from the first 3 exams. So your first 3 exams are going to be your study guide for half of your final exam. Right? So half of it's going to be cumulative. So please let me help you. Plus, again, as always, whenever you meet me online to get detailed feedback on an assignment, you will get a 5% boost to that grade. So that's it. I'm here to help. Just as always to remind you. So any questions about any of this stuff? Does anybody have their own announcements or reminders? All right. So today's first word for attendance is tag. When you tag somebody, apparently today is National Touch Tag Day. Oh, why? But it is. So that's our first word for attendance. Moving forward. Let's look at the big picture again. So again, we're now working on the stuff that's going to be on exam 3. Right? So not the stuff that's going to be on the exam that's due on Sunday, but the next exam. And for the most part, this exam 3, is going to be about reproduction in one form or another. Whether we're talking about mitosis, which is what we talked about last chapter, or we're talking about meiosis, which is what you're about to learn about, or once we dig deeper into it, then actually look at the mechanisms of how we're copying the DNA and things like that. In a sense, it's all about reproduction. But that's what we're doing. And again, we just got finished talking about mitosis, which is asexual reproduction, right? Now we're going to talk about sexual reproduction, and we're going to learn all about meiosis. And again, you can study however you want. My suggestion with this, I think it's usually best to make sure you've learned mitosis thoroughly. And then once you've learned that, then learn meiosis. Because in a sense, meiosis is almost like a variation of mitosis, right? So you know what you know about mitosis. So instead of me just teaching you meiosis from scratch, in a sense, I'm saying, all right, you know what you knew about mitosis? Well, it's just like that, except these little things that are different. So that's how I'm going to teach it. That's also how I recommend studying it. Anyway, what does sexual reproduction do? It reproduces offspring that have a combination of genes from the parents. In other words, if I were going to use the terminology that we use when we talk about asexual reproduction, it produces cells that are genetically unique, right? I think the term we used last chapter was that those cells were genetically identical to each other and the parent cell. Now we're saying that they're genetically unique. Or again, as this bullet point says, unique combination of genes, right? Different ways of saying the same thing. So, unlike asexual reproduction, sexual reproduction, which is what we're talking about now, depends on a process called meiosis, which is what you're about to learn about. Meiosis is the process that produces the sperm and the egg, aka the gametes. I'll break that word up again later. Right? That's it. That's what we use meiosis for, is to produce those sperm and eggs. Other than that, after you get fertilization, once that sperm and egg come together, after that, things are pretty much just as usual, as normal, like you've already learned. After that, mitosis comes into play. But Meiosis is all about producing that sperm and egg. So any questions so far? All right, just like the last chapter, I feel like the old textbook does a better job of teaching this subject. So because of that, these PowerPoints are still based on the old textbook, and therefore they're going to be a little bit out of order from your textbook. It's the same information presented in a different way. So I'm only saying this because if you're one of those people who likes to follow along closely with the textbook, which is a great thing, in this case it might throw you off a little bit. And don't be alarmed, it's all on purpose. So here's what we're going to learn about in general for this chapter. We're going to learn about homologous chromosomes. I've already started mentioning those, but not quite to this level. You're going to learn about gametes and the life cycle of a sexual organism. You're going to learn about the process meiosis. And then finally, we'll have like a review where we bring this chapter and the last chapter in together. We kind of compare mitosis versus meiosis. And then we'll go beyond that and learn about the origins of genetic variation and what happens when meiosis goes awry. And some of these, especially like this second last little point, that's going to be really important later in the semester. So you're going to learn about the origins of genetic variations, for example, mutations. So you're going to scratch the surface learning about mutations in this chapter. And then in future chapters, we're really going to dig into how mutations work. Then we're going to start talking about natural selection and other forms of evolution. And without these origins and genetic variation, without these mutations, we wouldn't have any of that. So this is all not just important for exam 3, but this chapter, especially that last bullet point, or second last bullet point. It's very important to understand the stuff you're going to learn later in the semester. So anyway, this is what we're going to learn in this chapter. Let's jump into it. The first thing we're going to talk about is homologous chromosomes. Here's something you already know, but I'm saying it again. Different individuals of a species have the same number and types of chromosomes, unless there's something wrong, right? So like I showed you that graph last chapter, it was from the old textbook that showed all the different numbers of chromosomes for all these different species. And I think a rat had, like a particular type of rat had the largest number, at least in that list. And I think there was some type of deer that had the smallest number, at least in that type of list. So yeah, very often the species has a very specific number, just like humans. As you need to know, this is the number you need to know. We have 46 chromosomes. That's one way of writing it. The other way of writing it, you need to know both ways. If we have 46 chromosomes, another way of saying that is we have 23 pairs of chromosomes. So sort of like if, I don't know, if you were at some kind of dating event, and there's 46 people here, 23 couples, right? Same thing here. 46 chromosomes, but there's 23 pairs of them. So you need to know that number. Now, where are these things found? These are found in something called your somatic cells. That's a term I might ask you about, but you definitely need to know what it is. I'm going to use it while teaching you this chapter, and I might use it in a question. Even if I'm not asking you what somatic cell means in the question, I still might use the term in a question. So make sure you know what a somatic cell is. And basically, a somatic cell is any cell in your body that's not a sperm or an egg. That's basically how you can remember it. Any cell that's not a sperm or an egg. Typical body cell. So you will find these 46 chromosomes, these 23 pairs of chromosomes in any somatic cell in your body, unless there's something wrong. Excuse me. Now, these 46 chromosomes, or 23 pairs of chromosomes, can be seen in something called a karyotype, which is this right here. So we're going to talk a little bit more about karyotypes. Now, granted, it's not going to be that important for the exam because there are few, if any, questions about karyotypes. It's also not going to be that important for the entire semester because even if you brain dump this information I'm about to give you about karyotypes, for the most part, you should be okay. Now, there are going to be some other smaller tidbits when I'm teaching you about karyotypes that are important, but karyotypes themselves, not that important. Also, again, since you guys are not biology majors, you're probably not going to deal much with karyotypes. But the last part of today's lab does involve karyotypes, so this will help you for that. So a karyotype is basically a picture of your chromosomes. To create it, what they do is they break open a human cell that's in metaphase and mitosis. And why do you think they do this in metaphase? Actually, let's make the question a little bit easier before we move forward. Why do you think we take a picture of these chromosomes when they're in mitosis? Why not take a picture of them when they're in interphase? Think about this. Try and take a picture of them. Think about all the pictures I've shown you of all these different cells in interphase and mitosis. Why do you think we want to do this when they're in mitosis as opposed to interphase? And again, forget the fact that it's metaphase, just mitosis in general. Why mitosis instead of interphase? Any guesses? This might be a good test question because there's nowhere in the book does it explicitly just say that. Well, I don't think it does, right? But what you know about interphase, what you know about mitosis, all that should come together and allow you to answer this question. No guesses?
Because they coil up.
Yeah.
So it's easier to see them.
Perfect, thank you, yeah. Just remember, if you send in words for extra credit, remind me that you got that so you get even more extra credit. But anyway, yeah, remember those pictures of interphase? It's just the nucleus looks like a big squiggly mesh. You couldn't tell one chromosome from another. And then once it goes into prophase and they coil up, they look like little sausages. So yeah, that's why we do it in in mitosis. But also, since we're trying to get pictures, obviously they never line up like this. So basically what a karyotype is we've broken it up, we stain the chromosomes with dye, and then we've taken pictures with the microscope. And then basically we do photoshopping and we arrange the chromosomes inside. So it's not like the cell ever looks like this, right? This is us getting a picture of all of those chromosomes with a nice and neat in the middle, right? And then we can kind of cut and paste and arrange them the way we want. That's how that works. Again, I'm not going to probably ask you any questions about that, but again, some of this is important, like that first bullet point. It's not important that you know when do they break open the cell to make a karyotype. It's important that you are able to think, like Stover did, and think, all right, well, oh yeah, the chromosomes are coiled up. When you take pictures of them, they should be coiled up. But yeah, that's how that works. So we arrange them from largest to smallest. And I think your textbook goes into this. I'm definitely not going to test you on this, but generally speaking, all humans, unless there's something wrong with them, again, almost definitely always have the same number of chromosomes, 23 pairs. But they're the same size, right? And not only are they the same size, but they generally have the same shape. I don't know if you remember what a centromere is. Here we go. I'll show you this one. This is a better picture. Remember the centromere is where these two sister chromatids are attached to that centromere. These two sister chromatids are attached to that centromere. So they all have the same centromere location too. So if you were to look at your karyotype and look at your chromosome number 1, the centromere would be here, and it would be like that for every other human. With some exceptions if something went wrong. Not only that, the genes are the same too. So say, for example, there was a gene that gave you a hairy back, and it was at the tip of chromosome 7. I'm making that up. I don't think there's one gene responsible for that. But anyway, if I had the gene for hairy back and it was right there at the tip of chromosome 7, that means if I were to look at your genes and look at your genome and look at your chromosomes, you have the gene for hairy back on chromosome 7. Of course, as you're going to learn later when we get into genetics, you might be like, oh, I don't have a hairy back though. That doesn't mean you still would have the gene for it. Your version of the gene would say, don't have a hairy back. So I should say a back hair gene for this hypothetical gene that I'm talking about. But anyway, all that is very important stuff that you're going to learn that's going to be important later. So again, the karyotype itself, not very important. But the fact that every human has like the same size and shape chromosome, centromeres at the same place, and all the genes at the same place, that is important. And notice again, for each one of these, there's a pair of them, right? Chromosome 1, you got this pair and this pair. You got, the one from your mom, the one from your dad. That is a homologous chromosome. These two are homologous chromosomes. So that picture of chromosome 1 is there's two sister chromatids, right? So this is the one, let's just say that's the ones from your mom, that's the ones from your dad. And of course, it's the mitosis, so they haven't split apart yet, right? So those sister chromatids are still together because it's a metaphase. But again, the idea here is for each one of your chromosomes, each 23 of your chromosomes, right, 46 total, 23 pairs, you have a pair of them. And for each one of them, you've got one from your mom and one from your dad. And for each one of them, the genes are the same. So again, if there was a gene for eye color, and it's not this simple, I'm really making this up. If the gene for eye color was in this little blue spot, the one that you got from your mom, right, that's where the gene would be in that little blue spot. That's also where it would be on the same chromosome that you got from your dad. Now, granted, you might have a different version of that gene from your dad, and we'll talk about that later when we talk about genetics. But again, it's very important that you understand what a homologous chromosome is. So you guys are familiar with homologous chromosomes, all right? Same chromosome, different version. You got your mom's, the one you got from your mom, the one you got from your dad. So a lot of this that I'm about to say is basically everything I just said, but now here it is in writing. So if you're more of a reading, you learn by reading, this will help you. Homologous chromosomes are pairs. Again, you get one from each parent unless something went wrong, and we'll talk about that later. They're the same length, right? The same shape, aka the centromere position. They carry the same genes. Now again, to reemphasize, this part will be later, report later. You might have different versions of those genes. Otherwise, you would be exactly like your mom and your dad. And that's not the case, right? So there's the same genes, but each gene has a different version of it. There's different versions of the same gene. But yeah, they do carry the same genes. And not only do they carry the same genes, the genes, and this is not written down here, but they're in the same location. Like on those hypotheticals I was giving you earlier. If the gene for back hair is on the very tip of chromosome 7 in my genome, it's the same in your genome. Again, unless something went wrong. So again, humans have 46 chromosomes, which is 23 pairs. These are numbers you need to know. Here's some new terms you need to know. Autosomes versus sex chromosomes. Sex chromosomes are pretty self-explanatory, right? Those are the chromosomes that, among other things, determine your sex. Sex, by the way, is different than gender. Sex is more of a biological term. Gender is more of a sociological term. And this is also, to point this out, this is a very simplified version of biological sex. So it actually gets a little bit deeper than what we're teaching. This is just the Cliff Notes 100 level version of this discussion. But anyway, sex chromosomes. For males, typical male would have an X chromosome and a Y chromosome. Typical female would have an X chromosome and another X chromosome. Those are your sex chromosomes. And all the other chromosomes are called autosomes. Not to be confused with autosome, like our place. Any questions so far? All right, so again, we have 46 chromosomes, 23 pairs of chromosomes. One pair of those chromosomes are our sex chromosomes. All the rest of them are called autosomes. And again, karyotype's not very important. Homologous chromosomes are very important. You're not going to be able to understand meiosis without understanding what a homologous chromosome is. You're not going to be able to understand genetics, which is the chapter after this, unless you understand the information on this particular bullet point. If you don't understand that, you're not going to understand evolution. So all this is very important as far as understanding things that are coming after it. So, any questions? All right, so that's it on homologous chromosomes. Now we'll move forward to talk about gametes in the life cycle of a sexual organism. So the life cycle of a multicellular organism is the sequence of stages from adults of one generation to the adults of the next, which is probably counterintuitive. I'm probably not going to ask you any things about that, but in the context of this conversation, when you think of the life cycle of a human, for example, you would probably normally think like I would think. So you're born and you die, right? But not in the context of this conversation. In the context of this conversation, when we talk about reproduction, right, sexual reproduction, then we're saying it's from the adults of one life cycle to the adults of the next. Because again, in this context, we're talking about sexual reproduction, so that's what we're concerned with. By adults, speaking of which too, in this context, I mean once you're old enough, once your body is able to sexually reproduce, at that point, in the context of this conversation, that's what adult means. So anyway, when we talk about this life cycle of cellular organisms, to be able to do all that, having two sets of chromosomes is a very important thing. Because as you're going to learn, and probably as you already know, and even if you didn't already know it, you could probably figure it out if you sat around and thought about it for a while. Since you have two sets of chromosomes, when you reproduce sexually, you're only giving up one of those set, right? You're like, all right, here's half my chromosomes. The mom's saying, all right, here's half my chromosomes. The dad says, okay, here's half my chromosomes. And together, you get a full set, right? So that's the key factor. This is all going to make more sense when I show you a picture, which will be a few slides from now. But are there any questions about this so far? All right, here's where it gets a little bit more confusing. I don't know if I want to use it confusing, a little bit more complicated. because all that stuff you probably already knew. Here's some new information. Some new words you might not know of that you need to know for the exam and for other stuff. Humans are what we call diploid dominant, which means that all body cells contain pairs of homologous chromosomes. So the second half of that bullet point is nothing new. You already know that all your body cells, your somatic cells, right, everything but your sperm and egg, you already know that all your cells have pairs of chromosomes, right? They have homologous chromosomes. The new thing here is that there's a term for that, and it's called diploid dominant. Your book also mentions diploid dominant and alternation of generations, but you do not need to know this. Know that humans are diploid dominant and know what diploid means. So there you go, that's a little note. That's what that little star is for. So when you're reading, you just need to know that humans are mostly diploid. Some people call it diploid. But what that term means is any cell that's diploid, that means it has two sets of chromosomes. That's all that means. It has both sets of chromosomes, right? Now there are some, well, I won't get there yet. So then we have, right, this is diploid dominant, meaning all of our somatic cells, every single cell in our body, unless something went wrong, has it has a pair of chromosomes, right? A set of chromosomes. We call that diploid. Now your sperm and your egg, that's a different situation. Like I mentioned earlier, they only have one set of each pair of chromosomes, right? So again, we have 23 pairs of chromosomes, so 1 to 23. For a diploid cell, or excuse me, a haploid cell, there's only going to be one of those from each. Only one of chromosome number 1, only one chromosome number 2, only one chromosome number 3. Now remember, every other cell has 2. And for each one of them, you've got one chromosome from your mom, one chromosome from your dad. And as you're going to learn later, there's going to be some astronomical numbers. These haploid cells are interesting because, again, they only have one chromosome from each pair. And whether it's your mom's version or your dad's version, that's completely random. We'll talk about that much later, but let that marinate for now. So yeah, those are haploid cells. Again, haploids don't have the pair of chromosomes, they only have one of each. That is what a sperm or an egg is. So make sure you know the difference between diploid and haploid. The way I remembered it when I was a freshman, the DI prefix always reminded me of 2 and half. That prefix, if I can speak, hap to me phonetically sounds like half. So I remember, oh yeah, haploid cells have half the number of chromosomes. So make sure you understand that. Anyway, just for your own knowledge, like I said, humans are diploid dominant, meaning most of our cells are diploid. There are some other organisms, especially like some really evolutionarily old plants, that are diploid dominant. Like if you were to look at all their cells in the plant, you would find that most of them are diploid. Excuse me, did I say diploid dominant? Anyway, some plants are haploid dominant, right? So if you were to look at all their cells, almost all the cells are haploid, but only a few of them are diploid, so they're kind of like the opposite of us. Not that you need to know that, just a little bit of trivia for you. You can look that up if you want to for extra credit. So I'm telling you that humans are diploid dominant, so you can look up what are some examples of some haploid dominant organ species. For that matter, what are some other diploid dominant species? Are there any species that are about half and half? Things like that. Anyway, so you know what diploid is, you know what haploid is. Now that you know those terms, we can quickly talk about the life cycle. In the life cycle of a sexually reproducing organism, a haploid sperm fuses with a haploid egg, and this results in a diploid zygote. You need to know those terms. There's definitely a test question that's going to sound almost exactly like the words that just came out of my mouth. Haploid sperm fuses with a haploid egg. The process is called fertilization. The result is a zygote, which is diploid. And again, that's exactly what we're after in sexual reproduction, is shuffling around those genes. Because once you combine those sperm and eggs, each one having half of a set of chromosomes, then once they come together, you now have a full set. So the next slide I'm going to show you is basically a visualization of what's on this slide, all the words on this slide. Are there any questions about the words on this slide? All right. So here's the picture. It's the cycle. Notice there's some curves right here. Everything's a big curve, right? I'm just throwing that out there because the next word for attendance is curve. So again, if you're watching the video, I should see a screenshot of me pointing at this particular curve. Curve is the next word for attendance. Apparently today is also National Curves Day. Based on the picture that I'm looking at, I don't think that means they mean that kind of curves. I think they mean that more in the aesthetic way, like how you're looking. But anyway, yeah, here's a picture of everything we just said. Right here, you have this haploid gametes. N equals 23, meaning how many? They literally only have 23 chromosomes. Right? That's what this means. These things only have. That one has 23 chromosomes. That one has 23 chromosomes. Right? Not 23 pairs, but 23 chromosomes total. Then they come together. Then you have this diploid zygote, which means it has all 46 chromosomes, right? So here N equals 23, right? The number of chromosomes is 23. So 2 times 23, right? 2 times N would be 46. Of course, that diploid zygote, that single cell, goes through mitosis and eventually becomes a full-grown human. And then that thing goes through meiosis to produce sperm and eggs, and then the circle of life continues. Now, I will say this. Your book talks about this, and you can learn about it if you want, but just keep things simple. When I ask any questions on the exam, I'm not going to use the terminology 2N equals 46 or N equals 23. Some people find that confusing. Since yours is a 100 level class and none of your biology majors, then I'm not worried about it. Just understand that a sperm cell and an egg cell, aka gamete, those are haploid, meaning now they have half the number of chromosomes, which for us is 23. And that the zygote and every other cell that comes after it is diploid because it has the full set of chromosomes. It has 46 total chromosomes. It has 23 pairs of chromosomes. And then know, as I'm about to teach you later in this chapter, that once you have this full-grown adult, then parts of it can go through meiosis and then produce those haploid gametes. And the next slide is actually going to be a very simplified version of how we go from diploid adult to haploid gametes. Again, just an overview. Then later we'll come back and really, really look at the details. But I like the way the old textbook goes, which is, hey, here's the big picture. Now let's start digging in and adding the details. Any questions about this slide? Okay. So here we have a pair of homologous chromosomes and a diploid parent cell. So imagine this was a human cell. If this was accurate, there'd be 23 pairs of these, but that'd be a little bit too much to draw and also a little bit too much to keep track of. So this particular organism is not a human. This particular organism has one chromosome. One pair, like a chromosome number 1. Of course, there's 2 pairs of it because it's sexually reproducing. So it's got one from its mom and it's one from its dad. So this is just a regular adult, 1 chromosomed individual, whatever kind of species it may be. Then it goes through meiosis, right? But before it does that, or excuse me, during meiosis, couldn't breathe. Before that, as we know from previous stuff, I'm going to write this down to also help remind you, because you're going to need to know this. We're not done with this. Remember when we were talking about mitosis last chapter? We mentioned before mitosis happens, during S phase of interphase, that's when the chromosomes duplicate. Well, same thing here, right? So we have this 1 pair of chromosomes, right? They duplicate, so now each one, the one from the mom is now sister chromatids. The one from the dad, sister chromatids, right? They've gone through that DNA duplication that you learned about previously. And in the later chapters, again, we're going to come back. Right now, that's the end of the discussion. Later, you're going to learn about all the enzymes involved in that and the whole process of going from 1 chromosome to sister chromatids. But anyway, that's how it first happens, right? We duplicated the chromosomes. Now we separate them in meiosis 1, right? So we've duplicated the homologous chromosomes before meiosis starts. Then during meiosis 1, we separate the homologous chromosomes. So for the exam, when you have any questions about homologous chromosomes, generally speaking, you should be thinking, well, definitely meiosis, because we don't even talk about them in mitosis. They're there, but they're not a part of the story, so we don't even bring them up. When you hear homologous chromosomes, you should be thinking meiosis versus mitosis. Specifically, because there's gonna be test questions, we have to differentiate between meiosis 1 versus meiosis 2. So when you hear homologous chromosomes, you should almost always be thinking of meiosis 1. Because the purpose of meiosis 1 is to separate those homologous chromosomes. So you've got one from your mom, one from your dad. After meiosis 1, you don't. They've been separated from each other. Now granted, look, those sister chromatids are still together. Nothing's happened there, but the homologous chromosomes are now separated. Any questions about that so far? All right, again, this is just an overview. As you might imagine, even though I haven't taught you yet, meiosis 1 also has prophase, metaphase, anaphase, telophase, cytokinesis, which is not, part of the mitotic phase. Anyway, that's meiosis 1. We'll talk about it in greater detail later, but again, the big picture here, the thing you should memorize before you memorize anything else, the purpose of meiosis 1 is to separate those homologous chromosomes. The one from your mom versus the one from your dad, they are now apart from each other. Then, as you might imagine, in meiosis 2, that's when we finally separate those sister chromatids, like you learned about mitosis. So in that sense, meiosis 2, in other ways too, as you're going to learn, meiosis 2 is a lot like mitosis. And we'll talk about that later. But again, big picture here. Meiosis 1 separates the homologous chromosomes. It's not until meiosis 2 that we separate the sister chromatids. Another thing worth noting, because this will be test question 2, is in meiosis, you start with 1 cell and you end with 4. In mitosis, you start with 1 cell and you end with 2. There'll definitely be test questions where you have to know the difference between those. In meiosis, you start with a diploid cell and you end up with haploid cells. Unlike in mitosis. In mitosis, you start with a diploid cell and you end with a diploid cell. Right, so these are things you definitely need to know. There's going to be a lot of test questions where you're comparing and contrasting the difference between mitosis and meiosis. And then also, there'll be some where you're comparing and contrasting the difference between meiosis 1 and meiosis 2. And again, the biggest thing so far is that in meiosis 1, you've separated these homologous chromosomes. Your parents' chromosomes that you hold inside your cells have now been segregated. And then again, in the second round in meiosis 2, that's when we finally separate those those sister chromatids. So that's it on our discussion specifically about gametes, the life cycle of a sexual organism. Next, we're going to move on to the process of meiosis. We're actually going to dig deep into this picture. Are there any questions about this in general before we start getting specific? Okay, so again, let's talk about the process of meiosis. Meiosis was when a germ cell divides and produces haploid gametes. So I've already said that before, maybe not in those specific words, but that's what I mentioned before, right? Of course, I didn't mention anything about germ cells. You can look that up if you want to for extra credit if you want. For this 100 level class, I don't care about that. Basically, what we're saying is it's not just willy-nilly. You can't just be any old somatic cell that turns into the gamete. There are specific cells. These are called germ cells. You can look into them if you want. I'm not going to ask you test questions about it. Yeah, that is what meiosis is, basically. Producing haploid gametes. Now, granted, again, I'm probably not going to ask you the definition of meiosis. Most of these questions are going to be more like comparing and contrasting meiosis with mitosis, and comparing and contrasting the two rounds, right? Meiosis 1 versus meiosis 2. And of course, a lot like mitosis, there'll be some questions where I say, in which part does this happen? In which part does that happen, right? Just like what I taught you in mitosis, it's going to be similar with meiosis. So again, big picture, meiosis is very similar to mitosis, but it has 2 major differences. You should definitely know these 2 major differences. The first one is that the chromosome number is cut in half in meiosis, right? In mitosis, we have a perfect copy of the original cell. Same chromosome number, same exact genes, right? Nothing changes in mitosis unless something goes wrong. In meiosis, we are cutting the number in half, right? It goes through 2 divisions. So it goes to that 1 S phase where everything duplicates as normal, like it does in mitosis. Then those chromosome numbers are cut in half in the first round when we separate all those homologous chromosomes. Then once again, we separate it again when we separate the sister chromatids. And because we have that 1 duplication followed by the 2 divisions, Each of those 4 daughter cells is now haploid. So again, in a sense, there's no new information on this slide. I'm just presenting it to you in a different way. Like, just to let you know, there's 2 major differences. You already know one of them, and this is it. Now you know it's one of the 2 major differences. Now I'm going to give you another one. This next one is new. I have not mentioned this. And this one will also be very important later on. There's something called crossing over. You know what crossing over is. It is the exchange of genetic material between homologous chromosomes. So again, for any given homologous chromosome in your cells, whether it be number 1, or number 23, or 17, whichever one it is, for any given chromosome number, you have 2 versions of it. You've got the one from your mom, you've got the one from your dad. That's nice and simple. This complicates things. Because what happens is when they get close enough together, and it just happens to be during the first prophase of meiosis, once they get close enough together, sometimes they literally swap chunks. Right? So then at the end of it, you could be left with a dad chromosome with a chunk of mom on it, and then of course a mom chromosome with a chunk of dad on it. That will be really important later in this semester to truly understand how genetics works. And that really does complicate things a lot. You'll see what I mean. I'm going to give you some astronomical numbers later. And those astronomical numbers don't even account for crossing over. Another thing I want to point out about crossing over is it doesn't always look like this, right? So it could be a chunk that size, or it could be a smaller chunk, or it could be a bigger chunk, or it could not happen at all, or it could happen more than once. So you could have a chunk here and a chunk up there. So the idea of crossing over, not a very specific one. This makes it look nice and clean. Like, oh yeah, that's what crossing over is. And it is. That's just one of billions of different versions of it, right? So that, again, the way they cross over can happen in so many different ways, so many different numbers of times. It's just a very simplified version. Anyway, to recap, this last slide here, right? There's 2 major differences between meiosis and mitosis. The first one is that the chromosome number gets cut in half. And the second one is this thing called crossing over. So any questions about the 2 major differences between meiosis and mitosis? All right, let's look at them. Let's look at these different steps individually. Again, excuse me, we have the interphase. As far as we're concerned, interphase prior to meiosis is the same as the interphase prior to mitosis, right? We're still dealing with the same thing. We have these uncondensed chromosomes. It's a big squiggly mess. The nuclear envelope is still intact because we don't have no need to disintegrate it yet. I'm not trying to move anything. We have these centrosomes getting ready, right? Because remember, that's where the mitotic spindle comes from. So they're gearing up to do their thing, but they haven't done their thing yet. That's interphase. And even though we can't see it, during S phase of interphase, these chromosomes have duplicated. They're just not coiled up yet, so we can't see it. Then, here we go. Prophase 1, very similar to mitosis, right? All the things you learned about mitosis are the same here. The nuclear envelope disappears, which makes sense, right? Because you're going to separate these chromosomes on different ends of the cell. So if you're going to do that, you need to get rid of the nuclear envelope that's keeping them together. Chromosomes are coiled up. Again, that should be intuitive. That should make sense too, because if you're going to move them around, you don't need a big squiggly mess. You need little things that you can move around. Speaking of moving around, the mitotic spindle is forming. And what you can't see here because it's not animated is that in prophase, not only is it forming, but they will eventually go ahead and attach to those chromosomes, those homologous chromosomes. So that's prophase, right? Everything's similar. Nothing's really much different between that and what I taught you previously. Now, here in metaphase, just like in mitosis, all these chromosomes are lined up in the middle of the cell. But in this case, again, I've already mentioned this, I'm beating a dead horse here, so hopefully you'll remember. Since we're talking about the first round of meiosis, it's not just like one long line of 46 chromosomes if that was, a human, if this was a human cell. Right now, it's the homologous chromosomes that have met up together. Right? The homologous chromosomes have met in the middle, not the sister chromatids, but the homologous chromosomes are lined up in the middle of metaphase 1. Other than that, everything's the same. Right now, we have this tug of war going on, just like we did in mitosis, trying to pull these things apart. But in this case, the thing they're trying to pull apart is the homologous chromosomes, not the sister chromatids. Once they are finally broken apart, right? Once that tug of war does its thing and they do break apart, that's when we have anaphase. Just like in mitosis, right? In mitosis, they've broken apart and they're moving towards opposite ends of the cell. Same thing's happening here, except of course now it's the homologous chromosomes that have been broken apart, moving across to the other sides of the cell. Not the sister chromatids, right? That would be mitosis. Here's the homologous chromosomes. Then while all this is happening, again, this is not animated, so what you cannot see is that the cell is getting longer and longer and starting to pinch off in the middle, because as anaphase is happening, also cytokinesis is starting to happen, and then it finishes it up at the end, right? We get this cleavage furrow. Again, it's not animated. If it was, what you would see at the end is 2 new cells. So again, everything's pretty similar for the most part, right? This should look a lot like mitosis. The only difference is here are these drawings. It's the homologous chromosomes that we've separated, not the sister chromatids. So any questions about what you see there? All right, so really quick, in writing, everything you just saw. Prophase 1, you just saw the picture of it. It's essentially the same as mitosis, except again, we're dealing with homologous chromosomes. We're not even dealing with the sister chromatids yet. The pairing, once they do meet up together, that's called a synapsis. I'll put a question mark next to that. I'm almost definitely not going to ask you what a synapsis is, but if I use the term in the exam, you need to know what it is. And of course, if you're reading in the textbook, it's going to use the term synapsis. So yeah, when those homologous chromosomes come together, we call that pairing a synapsis. Again, even though I didn't show it on that last picture, this is when crossing over occurs. That will definitely be a test question when this crossing over occur. Prophase 1 is what happens, which again should make sense because that's when they're together. Of course, they're also together in metaphase 1, but so that might be a little bit hard. But anyway. Oh yeah, again, as a point, just to point something out, I'm teaching based out of the old textbook. Your textbook is a little bit more complicated. It breaks down prophase and prometaphase. Right? So first there's prophase and there's prometaphase. So when you're learning that, if you're studying your textbook, just remember that for simplicity, to keep things easy in this 100 level course, I'm combining those. So whatever happens in prometaphase might as well be prophase. In short, all that stuff I taught you, yes, it happens in prophase, but if you were to get really technical about it, some of you would say, oh, technically that's prometaphase when I did that part. But you can look that up if you want. Again, I'm not going to get that complicated on the exam. So any questions about prophase 1? All right. And I don't even have any words about the rest of them. Because everything else was basically the same. So let's look at again, or not again. This is the second round of meiosis. So we're done talking about the first round of meiosis. This is meiosis 2. We've already separated the homologous chromosomes. So the only thing we're left with now are the sister chromatids. Now this should look even more like mitosis, right? We have these chromosomes ready to go. The nuclear envelope has disappeared. The mitotic spindle is forming, which you can't see because this is not animated, as eventually they do attach to those sister chromatids. And then they start moving towards the center of the cell. Once they reach the center of the cell, that is then called metaphase. Just like in mitosis, right? You've got these sister chromatids lined up in the middle for metaphase. There's a tug of war going on right now that you can't see because this is not animated. They're trying to pull apart these sister chromatids. Once they are pulled apart, we then consider that anaphase. This is not animated, so what you can't see is that not only are they pulled apart, but they're also moving towards opposite directions. And so is the cell. The cell is becoming more of a, or less of a circle, more of an oval. You can't see that happening. But anyway, this is what happens to these two cells. Eventually, they go through, they complete telophase, they complete cytokinesis, and what you have now is 1, 2, 3, 4 different cells. Right, 4 different haploid cells. Man, I really did get my slides out of order. Anyway, any questions about that side? All right, the next slide I'm going to show you would have made more sense had I had it in the correct order, so I apologize. So we just finished talking about prophase 1, meiosis 1. Then we went and talked about meiosis 2. And now, because I screwed up, let's back it up again and talk about metaphase 1. So sorry about that. So again, I really apologize. Bringing it back to the first round of meiosis, that's when we're dealing with the homologous chromosomes and not the sister chromatids. We had that one slide where everything in prophase was in writing, right? We had the synapsis and all that. So now in the writing for metaphase 1, again, this is essentially the same as mitosis, except It's not the sister chromatids that have met in the middle, it's the homologous chromosomes that have met in the middle. And this last bullet point is also really important for stuff you're gonna learn later. The way they line up in the middle is completely random. So it isn't as if all the remember these are homologous chromosomes, right? So for each one of them, you've got one from your mom, one from your dad. It is not as if all of them from your mom line up on one side and all of them from your dad line up on the other. It's completely random. So for all 23 pairs of your chromosomes, it is completely random. And that is what independent assortment is called. That's where I'm gonna give you those astronomical numbers that I keep talking about. Any questions about metaphase 1? All right, so that's it. Your last word for attendance, as always, even if I forget to say it, is going to be to ask me a question about the material presented in this in this slide. If you're in person, you don't have to send those words in, but if you do, you need to send them in by 9 a.m. to get extra credit. If you're online, you do have to send those words in. You have to do it before 9 a.m. to get regular credit. If you're watching the video, send them whenever you want, but don't forget to have the screenshots in addition to the words. That's it. I'll see you here for lab if you want to see me here for lab. Otherwise, I'll see you on Friday.